Okay. Hey. Can you close? Okay, so today is our last class, and I know you're very sad about it. I know I am, but if you're very sad, you can come next year also.
Do you want to first refill the final?
What?
To retake this class?
If you want.
Okay, let's make sure he's really done the final.
Yeah. So we'll start with the questions regarding the G protein coupled receptor chapter, and then we'll finish the last chapter of post and transcriptional Rna Regulation. So again, I'll tell you that we'll have a little time in the end, and we can talk, and you can ask whatever you want about the structure of the test and where to study from and stuff like that. And but in general, again, these questions, they're the questions that I give you in the end of each chapter are more or less at the same level that you'll see in the test. The only difference is that I invest much less energy in these questions, so they can be less accurate and have have a lot of segments and stuff like that. And in the test, I save the good questions for the test. So and I really take a lot of time in making the questions to be fair, and also absolutely, and I mean like in that they're completely loyal to the ground truth. Okay, like to the ground truth of what we know in science. So, for example, you'll not find a question in the exam that is not that if you even if you didn't take the course and you studied biology and you know that it's true, it will be true no matter what. So, okay. So the G protein coupled receptor, which was a big chapter, when the signaling molecules are received by the same cell that secreted them, the signaling is called autocrine signaling. What? Ah, okay. Which sentences are incorrect about G proteins? Okay, so mark all the sentences that are incorrect. For example, that kind of question does not appear in the test. So all G proteins activate their target effector proteins. Is that correct? It's incorrect. And the alpha subunit does not always facilitate the downstream effect. That's true. Okay. The trimeric protein is transmembranal. No, it's anchored to the membrane, but it's not transmembranal. Only the alpha subunit is capable of GTP hydrolysis. Only the alpha subunit is capable of GTP hydrolysis. That's true. Okay, like the beta gamma subunit don't have GTP hydrolysis capacity, so that's true. But it's a distraction because I say only. And a lot of times in biology, when you see only, it means it's not true. But in this case, it is. So the disassembly of the G protein requires GTP hydrolysis. No. So return to the scheme of activation, and the disassembly of the G protein does not require hydrolysis. It requires binding of the GTP, and before it's hydrolyzed. Later, it's being hydrolyzed by the alpha subunit. Okay, below are different steps of signal transduction pathway of G protein via adenylene cyclase in an arbitrary order. Arrange the steps in the order of occurrence. Ignore feedback processes. So what is the first thing that happens here out of these processes? G protein activation, then what? PKA? Increase in NA cyclase. What produces what produces cyclic AMP? So that NA cyclase activation, then the increase, then PKA, and then right phosphorylation, and finally RNA transcription is just one of the examples of the things that can happen. Many other things can happen. So GPCR signal transduction response is regulated by, or can be regulated by, changes in local concentration of protein components. Is that a possible mechanism of regulation of GPCRs? Yes, and we saw this example in the adaptation to light, where I showed you that the aristines and the G proteins change the locations, the cellular location. So this is a regulation of this process. Changes in the affinity between the G alpha and G beta gamma subunits. No. Phosphorylation of the transmembranous domain of the GPCR. There is phosphorylation, but there is no transmembrane domain. Not phosphorylation of the transmembrane domain. Okay, so here this is like a distractor, but finally endocytosis by the GPCR that happens after the aristine is binding. Last question: the aristine protein binds to G proteins and prevents its activation by a receptor. Is that correct? Does the aristine bind to the G protein? Binds to a receptor and prevents activation of the G protein? Is that correct? It is correct. Binds to a ligand and prevents activation of the receptor is not correct. And binds to DNA and prevents gene transcription? No. Okay, so this is gonna obviously for the test. I hope you'll know these things, because as you can see, it's pretty basic. Okay, so the last chapter that we talked about, and we divided it into. We're talking about the post-transcriptional control, or mechanisms of post-transcriptional control or gene control, and we separated it into three compartments: to the type of regulation that happens, or processing that happens in the nucleus, then talked a little bit about nuclear export, and now we're going to talk today about the cytosolic processes that happen in terms of regulation and processing of the mRNA. So the most basic step that happens to the mRNA after it's being transcribed is that a 5' cap is added to it, and this cap acts as a protection against degradation. The mRNAs that don't have a cap degrade very fast. The second modification that happens is addition of poly-A tail. This happens by the identification of a signal, polyadenation signal, somewhere in the close to the 3' or in the 3' untranslated region of the mRNA, and it involves many proteins and RNA binding molecules that know how to cleave the part of the mRNA that comes after the polyadenation signal, and then adding a multi-A segment. And finally, there are also proteins that bind to this poly-A, and we call it what? There are proteins that bind to this poly-A and stabilize this structure. So polyadenation binding proteins that bind to the structure and stabilize the poly-A tail. And both the capping and the polyadenation contribute to, first, the regulation of from the nuclear export, then prevention of degradation by exonucleases. So this is an example of exonucleases. that degrade RNAs either from one end or from the other end, and they promote translation. And like I told you, what we're going to talk about it more in depth soon, is that translation doesn't happen when the mRNA is in linear form. It actually happens in this closed loop structure. And the reason that this closed loop is important in regard to the poly A and the 5' cap is that the cap and the poly A bind proteins that know how to associate with one another. And only when they associate, they recruit the small unit of the ribosome that starts the translation process. So if there is no poly A or no 5' cap, the translation will not occur. Okay, so they have an active role in promotion of the translation. And actually, I owe you a movie that I didn't show you last time. So that summarizes these two regulation elements. You didn't see this, right?
Before mRNA can be used by ribosomes as a template for building proteins, it must first be processed. Key steps are the addition of a methylated cap and a polyadenylated tail. Involved in processing are RNA polymerase, cleavage factors, and poly-A polymerase. Processing of mRNA begins with transcription. Soon after RNA polymerase begins transcription, a methylated cap is added to the 5' end. Transcription then continues to completion. Following completion, RNA polymerase releases the capped strand of pre-mRNA. Specific nucleotide sequences in the mRNA are bound by cleavage factors. The 3' end of the mRNA is next moved into the correct configuration for cleavage. Stabilizing factors are then added to the complex. Poly-A polymerase now binds to the mRNA and cleaves the 3' end. The complex begins to dissociate, and the cleaved 3' end quickly degrades. Poly-A polymerase now synthesizes the polyadenylated tail by adding adenine residues to the cleavage site. Additional proteins then bind to the tail, increasing the rate at which it grows. When the tail reaches its full length, the poly-A polymerase is signaled to stop adding residues, and the polyadenylation process is completed. The processed mRNA is now ready to undergo splicing in preparation for translation.
Okay, so the next process is splicing, and this is a very nice simplification of what happens in splicing, where basically it's just when two portions of the mRNA, which are called exons, are fused together, and a portion that's called an intron because it stays in the nucleus. These are called exons because they are exported from the nucleus. So this portion is cleaved out, and this is a very basic mechanism that shows the actual process that happens here. Like somewhere in the introns there's an A that binds to this GU in the beginning of the intron, and then forms a lariat, and eventually the first exon here binds to this AG, or after the AG, and then you get the spliced mRNA here. Last week I mentioned a little bit the HNRNPs. I would like to talk a little bit more about them. Just to this image, for example, is a staining of a DNA molecule that is currently being transcribed. Okay, this is in a bacterial cell, so it's a little bit different from what we have in our body, but it's just in the staining that you see here, or the red staining that you see here, is an antibody that was targeted against one of these HNRNPs, which HNRNPs are special or small proteins that associate to RNA and assist in many of the functions that RNA has to undergo. And these images just to let you see what is the extent of transcription. Okay, it's not like when you saw before in the movie that you have this small ribosome that's attaching and making this nice little strand, and that's it. Often when there's transcription, there is massive transcription, and many copies that are made simultaneously, and many RNA polymerases that are running on the same gene simultaneously. And the mRNA molecule is not naked in the cell. It's covered with many RNA binding proteins, or HNRNPs, and also with small RNA molecules that create complexes together with these RNPs. So an example of this, of one of these HNRNPs, is RRM2, and this is just to show you, it has this is an image that shows the charge. Okay, so the relative charge. So RNA and DNA molecule, we talked a little bit about it when we talked about methods, actually, where a lot of people weren't present, but DNA and RNA molecules in general are negatively charged. So this protein has a vast region that is positively charged, and it attracts or binds in this way to the RNA molecule, and it facilitates many of the functions that the RNA or the mRNA has to undergo, together with polyadenylation, alternative splicing, and involved in transport. So basically, these proteins are doing all the works, all the things that we talked about and we're going to talk about are mediated by these small proteins that know how to bind RNA molecules. And also they're affected by phosphorylation. So you can imagine, if we connect it to what we saw before about the G protein coupled receptor, that the pKa can also phosphorylate them and cause activation and alternative splicing and all of the processes that we learned about. So I also showed you small nuclear RNAs, which are called snRNAs, or uRNA, are a special family of small nuclear RNAs that are involved in the splicing process. For example, the U1 snRNA is responsible of detecting the junction between the first exon and the intron, and the U2 RNA is responsible of detecting the branch point, the A that you saw before, that forms the array. And there is also there are many more of these uRNAs that are responsible for the splicing mechanism. So the spliceosomes, or in general in the splicing process, and here you see it in more detail than you saw before, the actual chemical phosphodiester bond that is forming and breaking. What's unique about this trans-esterification reaction is that it doesn't require any external energy. And I told you that this is one of the things that, when you see a molecule in biology that doesn't need any external energy to copy and paste itself, then it tickles your imagination to assume that maybe this had a very primal function, or this molecule was one of the first molecules that started life because of this property. For example, it didn't need to be in a cell. It could form spontaneous structures without the need of energy, so it doesn't need external involvement. We still see in some types of fungus, for example, we see some introns that are self-splicing and do not require any mediation and energy from external sources. We talked about the consensus sequence of splicing, like I showed you before, that there's G and U, and then there's this A and this AG, but in general it seems very redundant. Okay, so it's kind of strange, how this very loose sequence, not a very conserved sequence that can happen spontaneously, is something that marks the exons and the introns, but obviously I understand that it's a more complex regulation that happens here, and it involves many other proteins, including ones that know how to identify exons, which we talked about. I'll show you in a second. But in the end, the splicing itself in the eukaryotic cell is carried out by a complex that's called the spliceosome, that's composed both of these uRNAs and HNRNPs, or RNA binding proteins, and together they form this massive complex that knows how to splice out introns that are not needed. Okay, and yeah, this probably should have appeared before, but never mind. Anyway, so the mRNA is not completely naked. It's actually far from it, and each element of the mRNA, usually there are specific proteins that are called SR proteins that binds to exons, and there are the U proteins, the uRNAs and U proteins that bind to the segments of the introns, and probably together the binding or the combination of the binding of the different components marks for the spliceosome what exactly is an exon, what exactly is an intron, and what should be cleaved out, and what should be kept. And then we talked a bit about alternative splicing. So after we said that there is splicing, there are also forms of alternative splicing. So for example, a single gene and a single mRNA that can be produced from this gene can be alternatively spliced. So different elements, for example, in this example it's missing exon 3, and over here it's missing exon 3, and only have exons 1, 2, 4, and 5. And in this example it's missing exon 4. So and you can see that it produces different types of proteins. Okay, so a single gene can produce different types of proteins, and the exaggeration example, or the most I don't know how to say, the most exaggerated example of alternative splicing is with the gene DSCAM, which is short for Down syndrome cell adhesion molecule, that can have, because it has 95 alternatively spliced exons, it can have potentially 38,000 possible isoforms or possible different proteins. And we said that it got its name because this is one of the genes that are in chromosome 21, which is where the trisomy in Down syndrome occurs. What causes it to overexpress, and it's probably because the different isoforms of this DSCAM are what help different neurons know which neuron they need to connect to. Okay, so this very complex protein can have combination of different variants, can serve as a code for a cell, because it's a cell adhesion molecule. It means that it's expressing on the outer surface of the cell, so other cells can sense or can bind to this to that cell through this DSCAM, or identify the cell through the DSCAM, and probably it plays a major role in the correct wiring or synaptic wiring of the brain. And then I give you an example from the auditory system, where there is a special calcium-gated or calcium-activated potassium channel that is expressed in the hair cells that are responsible for sensing the movement of the basilar membrane of the hair cell, and it also has 8 alternatively spliced exons, and these 8 alternatively spliced exons can produce 576 possible different isoforms. Each one of them has a different sensitivity to calcium. Okay, so, and what's more interesting is that the decision whether to keep an exon or exclude an exon can be also affected by phosphorylation. For example, like pKa or other kinases can phosphorylate the HNRNPs and the SR protein that I showed you before, and alternate or change the form that will be expressed in that specific hair cell. What it means is that we saw that depolarization, for example, is one of the, we actually didn't see an example of that, but depolarization of cells is one of the things that can trigger G protein coupled receptor activation, which can lead to phosphorylation, which can lead to alternative splicing. So it's like, you can imagine it as this long-term feedback loop, that if a cell, for example, is activated too much, it will probably express a different variant of this channel that is less sensitive to calcium. Okay, so this is like a way, for me, it's like a way that the cells can fine-tune their sensitivity through alternative splicing. So also, when we're talking about alternative splicing, it has to come like in reference to what people in the past thought was the number of genes that we actually have, because people were trying to find things that are special or unique to humans. And because of that, in the 60s or in the 70s, people thought that we have 100,000 genes, and other animals have much less genes. But that's actually not true. And today we know that we have more or less 2,000 genes. So this is not what's special. So because, for example, a grape has 30,000 genes, and also plant cells are much more elaborate in the terms of the number of genes that they have than animals. And also, like a chicken has 16,000 genes, and a fruit fly has 15,000 genes. So it's not the number of genes that's probably special. But when you look at the amount of alternative splicing that happens in the organism, or the estimation of the number of the alternative splicing that happens, we see that humans have more cases of alternative splicing, meaning that they have the capability to modulate the variants of the different proteins better, or to a higher extent or higher complexity than other organisms. And this is especially prominent, or a feature that is typical of the brain. So in the brain there is always the most levels of alternative splicing, although I told you it's not very different from the liver and other tissues, so maybe the brain is not that special, but still. And I give you an example from our lab, that from an article that was published like 15 years ago or 16 years ago, when they found that Rna, who is a researcher here today, so we found that when you expose the mice to stress, then there's a shift or alternative splicing from the normal variant or the synaptic variant of acetylcholinesterase, which is the enzyme that degrades acetylcholine in the synaptic cleft, from this variant to this variant. Okay? So there is an alternative splicing to a variant they called it a read-through ACHE or ACHER. And this variant is special in terms that it has completely different membrane binding properties. For example, the synaptic ACHE binds to the synaptic cleft, and the read-through ACHE is diffused. It's not actually, it's released from the synapse, but it doesn't remain there. So it's much less effective in degrading ACHE. And this happens when and this is like in situ hybridization staining in the hippocampus of control animals and post-stress animals. And you can see this elevation in the read-through ACHE. Okay, then we finished with just talking one slide about the nuclear transport, just to show you that it's not like a simple pore, the nuclear complex pore. It has many proteins, and we're barely talking about it, but it doesn't mean that it's not important. And these proteins have major roles in regulation of transport of mRNAs from the nucleoplasm to the cytoplasm. And again, there are RNP proteins that are sitting in junctions where splicing occurred. And this is often used as recognition by this complex to allow the movement or the export of the mRNA from the nucleoplasm to the cytoplasm. So often if you have like a mRNA that was not spliced or didn't undergo proper splicing, it will not leave the nucleus, and hence will not be translated and will probably be degraded. Okay, so this is where we finished last time. And the last part of regulation is what happens to the mRNA when it leaves the nucleus and in the cytoplasm. So the first level of regulation that we're going to talk about is microRNA regulations. So microRNAs, you can say that they're special mini genes that are non-coding RNAs. They're part of the family of non-coding RNAs, meaning that they do not produce any functional protein, but their activity is in their RNA form. They're processed inside the nucleus. sometimes they have their own gene, and sometimes they're encoded inside an intron of another gene. But they undergo cleavage by special enzymes that are completely unrelated to the splicing enzymes that we saw before. They have an enzyme that's called drosha that cleaves the pre-microRNA, or you can say the mRNA of the microRNA before, and then makes it a pre-microRNA. Then this pre-microRNA is exported outside of the nucleus, and over there it's being cleaved again by a special enzyme that's called dicer. And this enzyme also loads it onto a special complex together with an enzyme that's called argonate, or AGO. So only one strand, in this case, one strand of the microRNA is active and loaded onto this complex together with AGO. And AGO, together with this microRNA, or this small, it's very small actually, it's like 20 nucleotides long, can either target cleavage of target mRNAs, can cause a repression, or can cause shortening of poly-A tail of mRNAs. Okay, so, and it's done according to recognition of special, what we call seed sequence, that is part of this microRNA. So there's like a segment of 7, 8, or 6, it depends who you ask, of nucleotides along this microRNA that bind to the target mRNA, and this is what causes the recognition of this AGO protein. So it's like, you can look at this microRNA as a mediator that mediates between this protein that's called AGO to the target mRNA, and when they bind to the mRNA, then it can cause either one of these responses. So because of the seed sequence is actually very short, it's only 7 or 6 nucleotides long, it means that combinatorically, and also we also see it happening in real life, is that a single microRNA can potentially target thousands to hundreds of target mRNAs. Okay, it's not that the same microRNA, but the same kind of microRNA can target many mRNAs, and also mRNAs can be targeted by multiple types of microRNAs. So it creates like a combinatorial regulation, or combinatorial suppression that one molecule can elicit on multiple mRNAs. The second type of post-transcriptional control, it's actually an artificial one, but it's important to learn it in the context of microRNA. It's called siRNA, and it's actually the siRNA is functional, or it's naturally occurring in plants and what we call lower animals, but we didn't see it, for example, in mammals, this mechanism. So the siRNA is very similar to the microRNA, but the fact that it doesn't have the seed sequence that I told you about before of the 7 nucleotides, it binds completely to the target. So it has 100% complementarity to the target, and it causes the cleavage of the target mRNA. So it doesn't cause like suppression or something like that, like I told you before, but it does act through the RISC complex, or the AGO that I showed you before, the AGO protein that I showed you before. And this time, because there is a full complementarity, the AGO just cleaves the target mRNA that it binds to. So I told you that it doesn't naturally occur, for example, in mammalian cells, but we use this as an experimental tool to target specific genes that we want to knock down. So for example, we can inject specific siRNA molecules inside, even give it to organisms or inject it into organisms, and it gets into the cells and can cause the suppression of specific genes or the degradation of specific genes. So it's more like an experimental tool, and the microRNA is more the natural occurring. So again, when you compare between the microRNA and the siRNA, so both of them are being cleaved in the cytoplasm by dicer. So either the microRNA that has this stem structure is cleaved to a double-stranded microRNA, or that the double-stranded RNA that we introduce artificially into the cell is being diced or cleaved by dicer to produce a double-stranded siRNA. Then only one strand is being loaded into the RIS complex, and the RIS complex is mainly AGO, what I told you, like the protein that I showed you before. And if we're talking about the siRNA, when you have full complementation, so it causes the target to cleave and degrade. And in the case of microRNA, because there is not full complementarity, only complementarity, here they show much more complementarity than what actually occurs, but it's mostly to suppress the translation process, but it can also lead to degradation, not only suppression. So here there's like I'm not going to go over this, but it's like a chart that compares between the two that you can go over later. They just summarize the differences between these two methods of regulation. And microRNA, for example, because again it's the more interested one because it's naturally occurring, can have all sorts of functions, including very interesting neuronal functions. For example, in retinal microRNAs, can play a role in light adaptation. So we see, for example, if we take the retina and measure the amounts of microRNA in the retina, so for specific types of microRNA, in this case microRNA 182, its levels are diminished in dark conditions and are elevated in light conditions. And this is relative to, so it's important for light adaptation because the control is a continuous light, so it remains like elevated all the time. And this is not the only microRNA that they saw. For example, and this is just to show you like how we call microRNAs. So for example, so microRNAs don't get names like other genes. They're normally just named with a short number and a letter, maybe. So you have microRNA 182, 183, 96, 211, and sometimes the microRNAs that were discovered in the beginning were called LET, so it's like LET7B, but still a microRNA. And in terms of synaptic function, if you, for example, take a cell or take a primary hippocampal cell or neuron and overexpress Mir125B, you see this phenotype that is elongation of the synaptic boutons of that cell, and you see exactly the opposite effect. If you overexpress Mir132, it causes the boutons or the synapses in the cell to be like short and stubby instead of long and thin. So this is only the effect, and this is just to show you the power of this regulation, that it has a significant effect, and this will obviously have significant effects on the neuronal function of this cell. And these two microRNAs are extremely important in terms of memory and other function, and have been studied extensively. Okay, so we're going to see a movie that summarizes the microRNAs and siRNA, and then we're going to take a break. Yeah? What?
It's regulated. So how could it be that light sensing is quite fast?
So you saw that it's like light adaptation. That it's so it's like light adaptation, and you see the time here. It's hours. So it's not like it's not seconds. But still, microRNA, one of the things about it is that it can act very fast. Like sometimes we think about regulation of the microRNA, of the mRNA, that is very slow, but actually it can happen, especially microRNAs can be transcribed very fast and be exported from the nucleus very fast because they're very short also, so it doesn't take a long time. And and they can be also regulated very fast. Okay, so for example, a microRNA that doesn't have a function or it's not loaded into the RIS complex, at that moment will be degraded very fast. So this can explain like what you see here. So you're right, but the kinetics is much faster for microRNAs than what we're used to in mRNAs. Okay. very dramatic movie.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy.
RNAi is siRNA, the same.
This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
Okay, any questions about that? No? Okay. So, we'll meet at 12. the other way, like writing the month before.
It does.
How?
Because here you would say, I don't know what this is, but you would say the first of the first, right?
And you would
So
say February 2nd.
Exactly. So the way you say it, it makes sense.
Yeah.
So it's the first time this ever happened to you, where somebody had a panic attack about a project due in a month.
Okay. By the way, you have the ability to walk in the dark also. Like you don't have to stay. If I would have known it in advance, I would just keep the lights off. Like each. Yeah. Okay. So, the last mechanism of regulation is more like in a specific example, which often happens in the ovum cell, before fertilization, in which the mRNA can be kept. If the mRNA is kept with a very short poly A tail, then it will be translationally dormant, so it won't undergo translation, and will stay that way if there are no enzymes that know how to degrade it, for example. So, and then if there's a signal that's coming, or some kind of polyadenylation enzyme that was upregulated, what it can do is it can start adding poly A tails to the mRNAs, and can cause the massive initiation of translation of these RNAs. And we see it, for example, in the ovum cell, that before fertilization, there's a lot of these RNA that are in silent form, and then upon fertilization, there's a massive overexpression of this polyadenylation enzyme that initiates the growing of the poly A tail of a massive amount of the RNAs. Actually, most of the RNAs undergo this massive polyadenylation, and this is what triggers the translation of these mRNAs. So, lastly, when we talk about cytoplasmic mechanism of post-transcription, obviously the concentration of the mRNA is a function of both its rate of synthesis and the rate of degradation, like we talked about in proteins. So the processes of degradation of mRNAs are very important for the abundance of the mRNAs. So, in different genes or different mRNAs may have different half-lives, and they don't have a constant life of that they're present before they undergo degradation. So, the way of the degradation, or the possibilities for degradation, are either from exonucleases. So first you have to have this decapping of the 5' cap, or removal or shortening of the poly A tail, which will lead to an exonuclease that can either degrade it from the direction of 5 to 3, or degrade the mRNA from the direction from 3 to 5, but it's not actually 5 to 3. It's actually a mistake. No, 3 to 5. Okay, 3 to 5 exonuclease that will degrade from the other end. And a more rare event, like we saw in the example of the microRNAs and the siRNA, is when you will have endonuclease cleavage, and this normally will not happen spontaneously. Where all mRNAs will undergo during their life this kind of degradation, either from the 5' or the 3'. Only ones that are regulated by, for example, for siRNAs and microRNAs and molecules like that, will be prone to endonucleases, and after the molecule is cut in the middle, it will obviously be degraded by the exonucleases from both sides. So yeah, I talked about this, and I talked about this. Okay, so the take-home message from this chapter is that in terms of regulation molecules, which are microRNA, splicing factors, HNRNPs, and all the molecules that we talked about, it seems that humans show a higher degree of complexity than other organisms in these molecules. And this is actually one of the things that I think is interesting for people that study non-coding RNAs, like we do in our lab, is that you really deal with the type of molecules that it seems to be very variable, or it seems that they have major differences in their abundance and complexity in humans relative to other species. And obviously the question is this what makes us special? Is yet to be determined. But it does look like we have this, that the level of regulation and the level of complexity is what we can say that is probably unique to humans, or the combination of all the different levels of regulation and complexity is something that is unique to humans, or more abundant in humans than in other organisms. And that's it. Thank you. So, see you in the test? No, a few words. So, about the test, there will be about, again, like I told you, I think before, I put also an example test. So you can see it on in the Moodle website. It's an easy test, okay? Which means if you know it, it's a test that I would, when I was an undergrad, I really liked these tests because it doesn't matter if you know or don't know, you're out in 20 minutes, okay? So it's like you don't have a lot to think about. It's either you know or you don't know. So it's very easy. If it's not clear, the papers are also included in the test. Okay, the two papers are also included in the test. I was asked here what is the level that you need to know about the papers. It's very hard to say that, but I will not ask you about supplementary figures or stuff like that. I will ask you only about things that we talked about in class, for example. And I don't really like to really say it's a rule, but in general, I don't think that there is an example, but don't catch me on the word that any question that will be in the test will be based on things that appeared at least twice, okay? But from what I see, from what I saw, like this is generally the rule that I take for myself when it could appear once, but I talked like half an hour on the slide, so it will appear. But in general, when I make the questions, then I look sometimes on my recordings, which is very embarrassing, but to see how much did I talk about it. And if I'm convinced that it's significant enough, then I put a question about it. In the test, 95% of the questions are 4-choice, multiple choice, not more than that. There are some true-false, and that's about it. If you have more questions, there are no choices. Everything you have to choose, you have to answer everything. And if you have questions, yeah.
About how many questions is it?
It's about 30. Each year it changes a little bit. Last year I think it was 33. So it'll probably be the same. Yeah.
Is it like evenly distributed between like the topics that we discussed, or is it like primarily focused more towards the later stuff?
No, it's evenly distributed in the time it took us to teach it. Like it's not. If like the G protein couple receptor, which is a large chapter, you'll have more questions.
And even like
Then
the basic first.
the basic first that we talked about a lot, you'll have.
Okay.
It's proportional. It's more or less proportional to the length that we talked about it.
Okay.
Okay, like for example, I think from the papers there's 2 questions on each paper or something like that. Like, but don't get out and leave if there's 3 or there's 1. Okay. So yeah, regarding normally I get requests, so I'm gonna offer it myself. I get requests for a rehearsal class, but I can tell you that in this course that it's not effective unless we do it a day before the test. Okay, so we can potentially, like, because you're gonna have questions, and and it's better to answer them together than to email me and then nobody sees like the response and stuff like that. Yeah, it doesn't work. Last year I tried that, and it didn't work. I can try that again, but I'm pretty sure it won't work again, because people will ask me on email and not in the forum. So yeah, so I'll schedule a time on Sunday. I see when one of the classes is available, either here or the 500s. But I will not come if there's like 4 people. So you need to send me an email if you're planning. I'll send you a message with today with the time and the place. Like, it not necessarily will be here, but if you're planning on coming, just write me. And if I get less than 4, then I'm not gonna do it.
Do you have options? Because on Sunday we have lots of classes.
I didn't look yet. I didn't look yet. So you're saying we can just so most people, if we do it like around 3 is okay for most of you, or not? No?
For how long?
Huh?
For how long?
Well, normally the length is dependent on you. Okay, I have no problem that it will be 10 minutes. But normally I stop it after 3 hours.
Sounds like Sunday for us.
Yeah, but you also you need to have time to study afterwards. But yeah, like I would follow the presentations and make sure that I know what's written in the presentation. Normally I don't have phrases there that I didn't talk about. Like, I remove them if I didn't talk about them. If it's there and you don't know what it means, then make sure you know what it means. And for elaboration and understanding a little bit more, you can go to the book. Although I told you that the book is more and more non-relevant. But for the first chapters and for the basic chapters, for example, the building blocks and the lipids and stuff like that, is very relevant. Like the basic biology. But for example, in this chapter, the post-transcriptional gene regulation, like I write chapter 12, but it's not like a lot of it is not relevant anymore, because it's this field is really progressing, like in science. So the book is not relevant in regard to that. Like, for example, like I told you, like
Bom lateral, Juninho.
no, I didn't know what was that. So like they say here that all these things are rare, cytoplasmatic localization that is rare, and all this stuff is completely not rare. Okay, and we know now that it happens all the time. In neurons, like I told you, like most mRNAs that are required for neuronal functions are differentially localized in different compartments of the cell. So it's not a rare thing. And we also know a lot about. I just told you like the classic cytoplasmatic mechanisms of post-transcription and control, but there are much more RNA species that have been discovered in the last 10 years, including long non-coding RNAs and circular RNAs and PIWI RNAs and a lot of different types of RNAs that just show us more levels and levels and levels of regulation. So, but it's not in the test. Okay, so make sure that you wrote your name in the attendance form. And so I'll try to schedule it to somewhere around 2, 3, something like that. Okay? אני אשלח לכם דרך המודל כאילו את השעה. לא יהיה משהו.
לא סיכמתי מספיק טוב בשיעור, נגיד המצגות הן מספיק.
תודה רבה.
אם את מבינה מה שיש להם, אז כן. לא יהיה כאילו משהו ש... למה?
לא, שלחתי לך את המיילים שלך.
כן, אז זהו, אז כן, שאלו אותי עכשיו, וביררתי אצל נירי, בגדול אני צריך להגיש את הציונים ב1 למרץ. בוא נגיד, אני לא יודע מה האורך של החודש של פברואר, זה בחודש הקצר, נכון? פברואר זה כמו 8. Do you know if February is 28th? Yeah. So, אז נראה לי כאילו משהו באזור 26 או 27 לפברואר.
אני מסיימת את המבחן ב-1 בבוקר, ואני יכולה לשבת. אני גם בשבוע שאני פנויה רק.
לא, זה לא צריך לקחת יותר מיום. את שואלת אותי?
אז אני אסתדר בזה.
רק בעניין של... רגע, בחרת מה אמרתי? זה הדבר הכי ש... כן. מקסימום נשלח לך אימייל כזה של... אם אני לא אזכור, אני כזה אגיד למה לא הגשת, אז תגיד לי דיברנו על זה.
תודה.
Oh, you want to help with that? That's almost production. Oh yeah? Yeah, it's done well. Oh, it passes well this week? I might also know this week. I've been like
וישלחו לי נקודה, אני רוצה לדבר חזרה. כי למה יש כל נקודה, נבחן אחד